All right, well, thank you very much, everybody. My name is Kirill Pankov. I'm a fourth-year BDC student, and today I'll be sharing with you the research I've done on cytochrome P450s, or CYPs, in the group of species that make up the Cnidarian phylum. Now, we're interested in cytochrome P450s because they play a large role in many important biological processes, from the synthesis of important steroids such as estradiol, metabolism of fatty acids to generate ATP for cells, and even metabolizing harmful drugs and foreign toxins out of the body. Now, these enzymes are found in all areas of life, including animals, plants, even bacterial species. And while CYPs have been studied extensively in humans and other model species like zebrafish and fruit flies, relatively little has been done in terms of CYPs in cnidarians. Now, there are more than 10,000 cnidarian species, including many types of jellyfish, corals, anemones, and other aquatic animals. And many of them have become cost-effective models for studies related to toxicology and other biochemical systems. Now, what's also interesting about these guys is they diverged from the rest of the animal kingdom 600 million years ago, and that makes them some of the most ancient animals alive today. This was before the development of bilateral symmetry, before the split between protostomes and deuterostomes, which defines so many of the species whose CYP content has been characterized. So in terms of invertebrate animals, they're way off in evolutionary space, and we're very interested to see how this superfamily has evolved in cnidarians, which key CYPs are absent, and how that's affected their development over time. Now, 3 different cnidarian species have had their genome sequenced that we're able to study, and each is interesting and unique in its own right, from Hydra magnapapillata, or the brown hydra, to Acropora digitifera, a species of stony coral, and the Matistella vectensis, or the starlet sea anemone. Now, collaborators of ours have done some preliminary work identifying CYPs in the Matistella, but relatively little has been done in either Hydra or the coral. So with this in mind, the goal of my thesis was to characterize the CYP complement of genes in these 2 cnidarians, using a variety of well-trained bioinformatics tools. Now, we start with what's often called the workhorse of bioinformatics, BLAST, or the basic local alignment search tool. It essentially tells us the parts of each genome that's most similar to CYPs from other species. We then manually annotate these significant regions by comparing to this model that we created for CYPs. It highlights the length of the sequence and also the presence of 4 key motifs that all CYPs have, each of which maintain the structural and functional properties of the enzymes. Now, we did this in both the Hydra and the coral, and here's what we found. There were only 18 complete CYPs in Hydra and 14 in the coral, meaning genes that are of the right length and have all 4 of those key motifs. Now, these are actually really small numbers and would represent the smallest known cypomes of any animal species. And while that's interesting in and of itself, our collaborators did very much the same thing in the Matistella, and they found 82 CYPs there, a significantly larger number. Now, we were surprised at first, but we were able to justify this apparent discrepancy by taking a deeper look at the history within the cnidarian phylum. Now, cnidarians can be divided into classes. So Hydra is in the Hydrozoan class, while the anemone and coral are both Anthozoan species. And even within this Anthozoan class, the split between those 2 cnidarians occurred about 500 million years ago. So there's still a significant period of time that each species underwent unique selective pressures, exposed to different environments, and that could very well result in a different CYP complement in each of them. Looking at these final numbers, it seems that extensive loss of CYP genes occurred in the Hydra and the coral, but not in the anemone. And while gene annotation gives us these total CYP numbers, it ultimately provides little functional information. So there might be 18 CYPs in Hydra and 82 in the anemone, but whether they're involved in metabolism of drugs or steroid synthesis or something else completely is still a mystery. But phylogenetics can help unravel part of that mystery for us. We first use a program called Clustal Omega to create a multiple sequence alignment of the unknown cnidarian sequences we found and many other CYPs from different species. The goal is to get a general idea which CYPs the cnidarian ones we found are most closely related to. So here we included over 400 sequences from many different CYP families and species. We then use that as input for a program called RAXML, which creates a maximum likelihood phylogenetic tree. It uses substitution models to visually show the relationship between our sequences. And this massive 400 sequence tree is shown here. Now, I know there's lots of different branches, but the important part is each color represents a different established CYP clan. The cnidarian sequences we found are highlighted in red, so the important takeaway here is the clan breakdown for each of the cnidarian species. That's summarized here, and a few things immediately stand out. First, the Nematostella clearly has the most diverse set of CYP genes, while on the opposite spectrum, Hydra only has representation from 2 clans. The clan 3 genes, which are the green portions of the charts, are a significant part of the Nematostella and Acropora cypomes, but they're completely absent in Hydra, so that means those could be a unique feature of the Anthozoan class of cnidarians I mentioned before. However, there are some limitations to this clan framework. Namely, while CYPs from the same clan are believed to share a common origin, natural selection has diversified the function within each clan greatly. So for example, some clan 2 genes are involved in steroid synthesis, others play a role in drug metabolism. And we can't pinpoint a specific function to a particular clan based on, or to a particular gene based on just its clan designation. However, what advantage of our analysis is that we can use this information to guide future experiments that will help test and answer these questions. So at this point, we've identified all of the CYPs in the 3 species, and their sequence and genomic location has been recorded. That means we can set up qPCR assays and other genetic tests to work with those genes. And the phylogenetics we've done provides us with a functional prediction, or at least a general idea for what each of them do. And that's all we need to set up new hypotheses that we can test with molecular work. So take CYPs and Hydra we think might be involved in steroid synthesis. When this species is sexually reproducing, it develops gonads, distinct bumps along the body. And we can isolate gonadal samples of Hydra and do qPCR tests to see if the CYPs we're interested in are being upregulated, since the gonads are a prime site for steroid synthesis. And that's just one example. Now, in humans, cytochrome P450s metabolize more than half of commonly administered drugs. We could do tests to see if the role of drug metabolism by CYPs and cnidarians is comparable to that in humans, and potentially even gather evidence to show that these species could be a good model for tests relating to drug safety and efficacy. There's lots of different avenues we can explore, and this will only improve as more cnidarian genomes become available. In fact, our lab recently received an early copy of the Aurelia aurita, or moon jellyfish genome. It's part of a 3rd class of cnidarians, the Scyphozoans, and inclusion of 3 different cnidarian CYP classes would make the phylogenetic analysis even more robust and allow us to gain more insight into cnidarian CYP evolution. This would send us further down the path of understanding how this superfamily has affected one of the most diverse and interesting groups of species on the planet. Thank you very much for listening. I'd like to thank my supervisors, Dr. MacArthur and Dr. Wilson, for all their guidance in this project, and also every other member of the MacArthur and Wilson Labs for their support over the past year. Thank you.